What we're going to do is just pick up where we left off, and then we'll go and give a general review. On Thursday, we were talking about just the phases of photosynthesis. We covered in really good detail the light-dependent reactions, and then what we concluded on Thursday was the light-independent, or you call the Calvin cycle reaction. So, in summary, where will the Calvin cycle take place? In the stroma of which organelle? The chloroplast. Okay. What is the starting molecule? Somebody else. What is the starting molecule for the Calvin cycle? Here. What is the starting cycle or starting molecule? Not for the Calvin cycle. That's for the light-dependent reaction. Carbon dioxide is an input. Very good. What else is necessary? That'll be necessary to drive it later. What is our starting molecule? Carbon dioxide will be added to it. I heard the word rubisco. Rubisco is actually an enzyme, so this will be used to carry out this process to carboxylate or add carbon dioxide. Remember, we're adding carbon to what? We're adding carbon to... Just look right up there. Carbon dioxide is added, by using the rubisco enzyme, carbon dioxide is added to RUBP, ribulose bisphosphate. If you remember, we said during the Calvin cycle, the whole point is we're going to take a 5-carbon molecule and add a carbon to it from carbon dioxide. Okay, so to go back and restate the question, it's like, what is the starting molecule for the Calvin cycle? And it's this molecule called ribulose bisphosphate. It's necessary, it's got to be present, and then we add which molecule before? Carbon dioxide. Okay, so we have a 5-carbon ribulose bisphosphate. We add a molecule of carbon dioxide to it. That new molecule has how many carbon atoms? 6. Okay? And we referred to it as just an unstable intermediate. This process of adding carbon dioxide to ribulose bisphosphate is called carbon what? Fixation. We're fixing, we're adding carbon to a 5-carbon molecule to make a 6-carbon intermediate. The enzyme that carries this process forward is called what? Rubisco. Okay. Rubisco, we said, has a really long name, so we just shorten it to something cute and easy to understand called Rubisco. Okay? So carbon fixation adds, the Rubisco enzyme adds what? Let's two molecules together, just to repeat. Rubisco adds carbon dioxide to something called RUBP. We can say RUBP. Ribulose bisphosphate. That produces something that has how many carbons? 6 carbons. That thing is pretty unstable, so it does what? Splits in to 2 separate 3-carbon containing molecules, okay, called PGA. Also, call it to the next molecule. So 2 molecules called PGA, but we need to add energy to this thing. We need to phosphorylate it. So which energetic molecules come into play here? ATP and NADPH. Where did the ATP get made? In this probably, maybe. Here we see it working in the chloroplast, so it was made following the light reaction using a molecule called ATP synthase. Which light reaction, which photosystem contributed to ATP synthesis? Photosystem 2, which actually happened first. Very good. All right, so ATP from photosystem 2 is added, system, which actually came from photosystem 1, which happened last. Okay? Once we use the ATP and the NADPH from our light reactions, a molecule called phosphoglyceride, or you can call it PGAL, PGAL, is made. And we noted that phosphoglyceraldehyde can serve as the template to make sugars like glucose, sucrose, fructose, etc. However, the bulk of it is used to do what? The bulk of our PGAL, or phosphoglyceraldehyde, is used not to create useful sugars, but to do what? To continue the cycle. Okay? And so the bulk of the phosphoglyceraldehyde molecules actually consume more ATP from that was made during the light reactions, okay, photosystem 2. Okay? So we consume more ATP as a result of photosystem 2. And then we help produce this molecule called ribulose bisphosphate, which was actually the starting molecule for the whole Calvin cycle. And so the takeaway I think you guys walked away with on Thursday is this is a very expensive process to make sugars to help for that plant's or that organism's nutrient metabolism. However, the bulk of the energy input is actually used to just keep the cycle going, to make a modest amount of phosphoglyceraldehyde that is a template for sugars like glucose, sucrose, fructose, etc. What questions do you have at this point over the Calvin cycle? Where it occurs, the inputs, the outputs, the purpose?
So one of the reactions of the Calvin cycle is
Yes. One of the reactants of the Calvin cycle, the first reactant, is ribulose bisphosphate. That's right. And also carbon dioxide. Okay, so the initial reactants for the Calvin cycle are ribulose bisphosphate and carbon dioxide. They're anabolized together using which enzyme? Rubisco. Do what? Nabisco. Rubisco. Ribulose bisphosphate. Not ribulose bisphosphate. Rubisco. This is the rest of the name starts out like that. Additional questions at this point? Alright, so do you think all organisms, bacteria, plants, corn in the field in the summer, pine tree in the winter, plant over in Australia, do you think they do everything exactly the same? No. There are variations in here. The cycle which we just described is called the C3 pathway because we produce how many carbon containing molecule? 3 carbon containing molecule. But there are variations in this cycle. We already went through this last week. If you look at figure 5.13, it compares the different types of pathways with a C3 versus what's called a C4 versus a CAM plant. Okay? So if you look on page 7 of your chapter 5 handout, we're just summarizing these events, right? Comparing what's called a C3 versus C4 versus a CAM plant. Not all Calvin cycles start exactly the same. So if you look at the C3 plants, we said these happen where? These reactions occur. So in summary, these reactions, the Calvin cycle occurs in that. Physically, the Calvin cycle occurs in the stroma. And what part of the leaf? What was the name of those cells? mesophyll cells. Okay? So in mesophyll cells, and more specifically chloroplasts in the stroma of the mesophyll cells of leaves. The inputs for the C3 pathway, we said, I'm going to cover that up, it includes what? Carbon dioxide and RUBP, ribulose phosphate. Also in that table, I have a note about what's unique about this particular pathway. And what's unique about it, you can see here listed as a limitation, is this concept called photorespiration. So just right there, what's unique about it is what's called photorespiration. Photo, P-H-O-T-O, respiration, R-E-S-P-I-R-A-T-I-O-N. Photorespiration. When carbon dioxide is not available, so think about when a plant has to close its stomata because it needs to conserve energy, something like water more specifically. It can use its byproduct oxygen to carry out an alternative pathway. Okay. The disadvantage to this is, well, the advantage is the plant can still make sugar, but it's going to make what? What do you think? More or less? Less. So it's a backup pathway with carbon dioxide. When carbon dioxide levels are low, relatively low, the plant can use oxygen for an alternative pathway. I'll show you in a minute to better support this. Okay? But right now we're saying a uniqueness to this is what's called photorespiration, where not carbon dioxide, but what can we use? Oxygen in extreme conditions. The next row that I have, it says used by percent of plant species, and this is a good guesstimate right here, about what percentage of plants utilize this pathway, the C3 cycle? About 95%, okay, so the bulk, right, that's why the textbooks tend to hone in on what's called the C3 pathway, because almost all plant species utilize this pathway. It's a common, unique path, not unique, but common pathway to many plant species. I already talked about photorespiration. We'll come back to that in just a minute. And typical habitats, just areas where there's plentiful moisture. Okay, so where you're not looking at dry, arid type of environments, where there's plenty of moisture available. Okay, so the types of plants that you're familiar with seeing here in Oklahoma use which pathway probably? Most of them use the C3 pathway. There's a C3, there's something probably else has a different name, and there's another set of plants that use what's called the C4 plant, and their intermediate product must have how many carbons? 4 carbons. Okay. This pathway starts in the mesophyll cells. Okay, so if you write on this what's a location, it starts in the mesophyll cells where we're familiar, where carbon dioxide is converted into a, how many carbon containing molecule? 4 carbon molecule. So the C4 pathway starts in the mesophyll cells where carbon is fixed and a 4 carbon molecule, not a 3 carbon molecule, intermediate is produced. Yes. In the C4 plants, atmospheric carbon dioxide enters in the mesophyll cells where it's fixed and a 4 carbon molecule is produced, not a 3 carbon molecule. That 4 carbon intermediate molecule is then transferred to a neighboring set of cells called bundle sheath cells. So again, that 4 carbon intermediate is then transferred to to another set of cells called the bundle sheath cells. They're right next to, they're right adjacent. And that starts the Calvin cycle. The 4-carbon molecule in the bundle sheath cells initiate the Calvin cycle. So it's a different set of cells and a 4-carbon molecule instead of a 3-carbon molecule. Different location, different number of carbon atoms, but the process is similar. Still trying to make sugars. That is called the C4 pathway. The limitation is it's a lot more expensive, even more expensive than the C3 pathways. And so the problems, I guess I can pop down to the bottom, is it's expensive. It's energetically expensive, or more expensive. What's the typical habitat described here? Where it's hot and dry. Classic example of a C4 plant shown in the image here is what? Corn. Okay? That's corn. So think about when corn grows. It grows in the summer when water levels are usually a little bit less. It is definitely hot. We can see that plant certainly growing here in Oklahoma. So that's a good example of a C4 plant. So they've got some specialized leaf structures to trap that carbon dioxide and prevent water loss. So they're trying to prevent water loss. And the reason it's got these extra steps. What percentage of species utilize the C4 pathway? What, about 1%. So not very many, but kind of like them. They make good tasty food, for example. So the C4 plant, it still carries out the Calvin cycle, but this time the Calvin cycle doesn't occur in the mesophyll cells. Where does it occur? In a set of cells called bundle sheath cells. And to start the pathway, there's a 4-carbon intermediate molecule that initiates it, yes.
So it starts in the bundle sheath cell?
The Calvin cycle occurs only in the bundle sheath cells. I kind of started intermediate, right? So the whole process originates in mesophyll cells. So I can maybe be more clear on my question. The Calvin cycle definitely starts and finishes where? In the C4 plant. In the type of cell? Bundle sheath. But carbon fixation occurs in the mesophyll cell.
No, I was just
So I could have been more clear. The Calvin cycle specifically in a C4 plant takes place in the bundle sheath cells. However, carbon fixation occurred in mesophyll cells. There's another series of or type of plants that can also carry out the Calvin cycle, and they utilize the CAM pathway. And these cells, these types of plants still utilize which type of cells? Mesophyll cells is where the Calvin cycle will still occur. But notice some features about this. What type of plant is shown here? Like a cactus, so succulent plants. Where do you find these types of plants? Deserts, where dry, hot, daylight hours long, water levels less in the environment. And so these plants have adapted, evolved, right, to survive in these environments. And so that we still have which gas? Carbon dioxide is still the common source of carbon dioxide. Notice, though, we still form not a C3, but a C4 molecule that initiates the Calvin cycle. Here's what is unique about this. When is the carbon dioxide trapped fixed? During the night. Because what should those plant cell stomata be doing during the day? happens is, in these types of plants, during the night the stomata open, carbon dioxide enters, and it gets trapped and stored. It just gets stored in this 4-carbon molecule. Because the plant can't store carbon dioxide, but it can store it trapped something in the 4-carbon molecule. So during the day, the 4-carbon intermediate accumulates, just hanging around, and when the light's available, the cell can do which reactions? The light reactions, which produces ATP and NADPH, and then during the day, it can carry out the Calvin cycle because it has reserves of carbon. About what percentage of the plant species utilize the CAM pathway? 3 to 4%. So still a modest amount. But certainly I think you guys can appreciate that not all species utilize the exact same pathways. There are some variations. But they're still able to adapt to their environment and survive. So the big thing is the heat. Just like what happens is it shouldn't get so cold that the inside of the plant would freeze and die. I mean, it could happen, certainly, but normal ambient temperatures, it won't get that cold for the plant to freeze. However, during the day, if the plants were to leave their stomata open, all their water would evaporate, and then they would die faster in that case. And so it's more of a heat excessive than a heat lower limit for the plants in the deserts. But you're right, there's big, crazy extremes. It's like Oklahoma. All right, so if we look at, I was going to walk through that. Figure 512 in your textbook, it illustrates for you some different types of leaf cell structure. We already went through the role of the epidermal cell and layers in the mesophyll cell. If we look on the far left, we can see a C3 plant. We can see a nice mesophyll cells. And then we haven't really talked about the transport of nutrients from one spot to another in the plant. But I think you could understand if the nutrients are made in the leaf, they aren't stored there. They're transported to be used somewhere in, like, say, a flower or in the fruit. Okay, somewhere they're going to be stored somewhere. Okay, so there's got to be a transport system. And those are here illustrated as the vascular tissue. And so the bulk of the photosynthesis occurs in these mesophyll cells. We also noted the role of the stomata that can mechanically open and close in changing temperature environments. If you contrast that, though, you can see a big difference between the C4 plants. You can still see some mesophyll cells adjacent to the stomata. But notice how large these cells are around, well, these are the mesophyll cells too, around the vascularity. So there's another set of cells large in the C4 plants, where we noted in the mesophyll cells, the carbon dioxide is fixed into a 4-carbon molecule that is then physically translocated into what's called the bundle sheath cells. So these little kind of almost circular looking cells would be called the what? Bundle sheath cells, where which reaction occurs in C4 plants? Not carbon fixation, the Calvin cycle. In these plants, carbon fixation occurs in the mesophyll cell. So, to restate that, in the C4 plants, the mesophyll cells actually just fix the carbon to form a 4-carbon intermediate, which is then transferred over to the bundle sheath cells that actually carry out the Calvin cycle when conditions are ideal. go back to that description of photorespiration. So if you look in your C3 plants where it says problems that have just the word photorespiration, if you look at this image, this is illustrating what we've understood in C3 plants in the subset A, where carbon dioxide is fixed to ribulose phosphate by the enzyme called rubisco to form first a 6 then 3 carbon phospho PGA molecule. Okay? So the first image that I have shown is what you're familiar with. However, we noted that when carbon dioxide is low, oxygen can be utilized to carry forward the reaction. Okay? So rubisco, right, can combine ribulose phosphate and oxygen. Notice the difference gas, right, in the second scenario. Instead of getting 2 3 carbon molecules, we get 1 3 carbon and a 2 carbon byproduct. So the carbon containing molecules are less, but look what happens. When that 2 carbon molecule is available, it can liberate a little bit of carbon dioxide, which could fix. Okay, so in extreme conditions, photorespiration can occur, but the amount of glucose produced is going to be lower, and the ATP inputs will be higher. But the plant still lives, and it still makes nutrients. And that's called photorespiration. We're to summarize photosynthesis. Oh man, I'll just skip that because that's not showing up too well. Let's just summarize this. This is the on page, should be page 103 of your textbook, to summarize photosynthesis. We said photosynthesis occurs in 2 stages, and you can label it on this handout if you want, on page 7 of your handout. Okay, there are 2 stages of photosynthesis. The first is the light reactions. Okay. The second type of reactions are the carbon reactions, or you can say the, what word have we been using? What term? Light independent, or collectively called the Calvin cycle. Okay. The source for the light reactions the source of electrons for the light reactions is water. Okay. The energy source is for the light reactions is what gives energy to the light reactions? Sunlight. Okay. Sunlight. So if you remember, in order for the light-dependent reactions, the plant will need obviously light, it's dependent upon light, and water to give electrons. And described here as a waste product, or maybe more appropriately, you can call it a byproduct, is what? Oxygen. Okay, definitely a byproduct. Which we'll learn, be used in the mitochondria, chapter 6, in that plant, or liberated out into the atmosphere. Okay? Out of the stomata, out in the atmosphere. But we said the big deal with the light-dependent reactions is to produce the energy source. What photosystem 2 produces for us? ATP. As well as photosystem 1 gives us electrons in the form of a molecule called NADPH. Okay. Those provide energetics and electrons for the carbon reactions of the Calvin cycle. We notice in order for this to happen, carbon fixation is necessary, so our source of carbon dioxide, carbon can do this, is which molecule? The carbon actually comes from carbon dioxide. The electrons come from NADPH. The carbon source is carbon dioxide. The whole point of that is to produce what? Glucose or other types of sugars. Yes, ma'am. glucose is what we'll study better in chapter 6. Okay? The whole point of that is, well, it can be used to release a different form of energy for the plant. So it can be catabolized. It could be used to make a cell wall, because you can remember plant cell walls are polysaccharides. It could be used to be stored in the cell in the form of starch, right, for later, that we would consume. It could be used to power aerobic respiration. Okay, so there's lots of reasons why that glucose needs to be made. Good question. Additional questions? This is summarized for you in your study area and at the end of chapter 5. So if you don't have not written all this down, you've got it for you in the end of chapter summary. Okay, putting it together, something like that. On or around page 103. Okay. Let's fill in this little handout that I have for you in the chloroplast. Again, it's repeating what we just walked through. With repetition, hopefully we'll start to get it. So summarizing the light-dependent and light-independent reaction. In order to start the light-dependent reactions, first of all, what organelle is this? Chloroplast, okay, inside of a plant cell. That's what we've been studying. Source of electrons, step number 1 is from water, okay? We said electrons come from water, energy comes from point 2, which is light. These collectively are going to drive what? These processes we call the light reactions. Okay, they occur in the what? Thylakoids. Okay. The light reactions occur in the thylakoids, and a byproduct liberated out of the chloroplast is a gas called oxygen. We care about that a whole heck of a lot because that's what we use. But for the plant, it cares not so much about the oxygen production as the what? 0.5. ATP and 6 NADPH, which are going to drive the Calvin cycle. In order for the Calvin cycle to occur, an atmospheric gas called what has to be available? Carbon dioxide will enter. It is fixed to a molecule called RUBP, ribulose bisphosphate, using an enzyme called Rubisco. Okay. These reactions occur, and I'll come back to that one. I'm missing something. And produce for this plant what? Step 9. Different types of sugars like glucose. Okay. However, when it produces this, NADPH, when it becomes oxidized, it forms what? NADP, ATP, yes, and ATP is going to be phosphorylated to form ADP. NADPH is oxidized to produce NADP charge. ATP is dephosphorylated to form ADP, adenosine diphosphate. These reactions occur in this chloroplast stroma. New questions after this. Sign-in sheet. What was the question? Yes. When you go to your Connect site where it says My Lectures, open there and you have to make sure all your pop-up settings are turned off and those kinds of things. And so, yes. All right. What I want to do is go ahead and transition to Chapter 6. Okay, so if you don't have your Chapter 6 handouts, of course, I would recommend taking just traditional notes. Okay. All right, so we're going to go ahead and transition to Chapter 6, which is dealing with the specific reactions inside of an organelle called the mitochondria. Okay, so certainly these two chapters' content go hand in hand, following up the energetics presentation. And so in chapter 5, we basically see how glucose is made, and in chapter 6, we're going to see how glucose is used. And so they're very much interrelated with one another. The chapter objectives will be shown on your your handout when you go into detail and check it out. We're going to talk about how converting that chemical energy like glucose into what's called kinetic energy. We're going to talk about the overall reaction in aerobic respiration, which should look familiar. It'll look like the reverse of photosynthesis. We're going to talk about 3 important, but not all of the metabolic pathways. We're going to start out with this process called glycolysis, the Kreb Cycle, and a familiar term called the electron transport chain. So a lot of the reactions will look kind of similar to photosynthesis. We'll talk about where 2 of the reactions occur within a mitochondria, and then we'll go in better detail the glycolysis or glycolytic pathway, the Kreb cycle, and the electron transport chain. We'll talk about where oxygen is specifically utilized, and then we'll get down to the numbers, like seeing how much ATP can actually be synthesized following each of these reactions. On Thursday, we'll talk about some alternatives besides sugar to the aerobic respiration, which you can think about what you might need for a balanced diet. Do we need to eat only sugar? No, it would be good. We also need proteins, and carbohydrates would be sugars, and also what are our other macromolecules that we probably need to eat besides proteins and sugars? Fats, okay? And so we'll see, well, how do they actually relate into ATP synthesis? Also, if we have time, we'll talk about, well, what happens if oxygen's not really available, okay, which is called anaerobic respiration, and then fermentation, and then we'll tie in together aerobic respiration with photosynthesis. Probably won't talk about evolutionary links, but certainly a theme in there. You ready to roll? Sorry. No. All right, so what we're going to say is, in order to survive, we know from the introduction to this class, cells need a constant input of energy. Okay, and that's what we're going to study. Where does this energetic come from? We studied in chapter 5, photosynthetic organisms get their input of energy from the sun. Okay, so sunlight is where the energy comes from for photosynthetic organisms. For organisms such as yourself, your input of energy is food, right? So we've got to eat food. That's how we start our energetic transformation. We're going to study 3 pathways. They're certainly not the only 3, but these are 3 common, really important pathways. So that's why we're going to select 3. If you're curious as how many pathways there are, you can come down to my physio lab, and I have a big 3x2 folder or poster to summarize all the metabolic pathways and the variations to this. So it may seem like we're learning a lot, and we are, but we're not learning everything. We're not learning all of the details. Okay. And so to summarize for you, the 3 pathways that we're going to study, or types of pathways, include what's called aerobic respiration, anaerobic respiration, and fermentation. Aerobic respiration, anaerobic respiration, and fermentation. What do you know about aerobic respiration? It uses oxygen. So in aerobic respiration, oxygen is used, and I want you to clarify this. Yes, oxygen is used, but what it's used for is to accept electrons. In aerobic respiration, oxygen is the electron acceptor, is the final electron acceptor. So what you're adding in here is that in aerobic respiration, oxygen is the electron acceptor in the pathway of aerobic respiration. As a result, following the pathways, carbon dioxide is actually released. I repeated it. I'm going to repeat it again to make sure you understand. In aerobic respiration, oxygen is what? Is used to accept electrons. Doesn't indicate it just has to be present. It's actually present and what? Working. Used. Okay. Just because something's present doesn't mean it's always used. Okay, so I want to make that clear in the other pathways. Oxygen may be present, it's just not used in the remaining pathways. So in aerobic respiration, oxygen is not only present, it is used, okay, to accept electrons. And then carbon dioxide is liberated. Anaerobic respiration must then what? Not use oxygen to accept electrons. It uses something besides oxygen to accept electrons. So it utilizes something else besides oxygen to accept electrons in this pathway. In the third pathway, you're like, well, fermentation, wouldn't that fit maybe into anaerobic respiration? In fermentation, it's most specifically NADH, which looks familiar, doesn't it? Sounds like NADPH. It's very similar, except it doesn't have a phosphate group. In fermentation, we know which molecule is utilized for electronic transfer, and it's NADH is used for electron transfer. NAD does, and when it's reduced, it's formed to NADH. So in the form NADH, it is not charged. It's oxygen. In its reduced form, it's neutral, and its oxidized form, it's charged. It's positive. Yes. Good observation. So aerobic respiration, what? Uses oxygen. Or anaerobic respiration does not use oxygen, use something else. And fermentation uses most specifically NADH. And we'll look at those details on Thursday. We're going to focus first on aerobic respiration. This formula should look kind of familiar, and it should look like the reverse of photosynthesis. And so if it's the reverse of photosynthesis, we must be starting with the input of glucose and oxygen, and releasing carbon dioxide and water and energy. The energy is not, we're not releasing like photons, but the energy being released is a good molecule, useful molecule called ATP. So, we're going to take an input of glucose, input of oxygen, and transform it to produce ATP, which then can drive other reactions and liberate water and carbon dioxide. This is the overall simplified reaction for aerobic respiration. Yes? Yes, they're important. This is just a molecular formula for glucose. If you remember, our carbohydrates have always a 1 to 2 to 1 ratio for carbon, hydrogen, oxygen, and glucose is easy, and that is just 6, 12, 6. So that's the molecular formula for what? Glucose. C6H12O6 is just glucose. This is just oxygen. This is just carbon dioxide. This is just water, and now you're familiar with ATP. So I think, yes, and I think once you kind of step back and after the panic of that formula wears off, it's a lot more approachable. So once the trauma wears, just think of the reverse. if I were doing photosynthesis, I need energy and water and carbon dioxide, okay, to produce oxygen and sugar. And this one just drives it backwards. Figure 6.1 has a nice little visual summary of what the purpose is. And we're putting, a good example is just when you exercise, whether you're actually going to the gym or just writing, taking notes, right, whatever. You're going to take in oxygen, exhale carbon dioxide. We know that. Now we're getting into the nitty-gritty. What happens? Well, oxygen we're going to see is getting transported into your cells, and most specifically in your mitochondria, to do what? To what? What do we, in the introduction I made an emphasis oxygen is used to accept electrons. The whole point of the reason you need oxygen is not to go round and round in your blood, but to accept electrons in the mitochondria. It's going to take a long time to get there, though. There's a whole bunch of reactions. Because we want to do this slowly, because why? We want to be, if I do something more slowly, I'm probably going to be more accurate and efficient. And so we're going to see all these reactions occur. It's going to take a while, but the whole point is it's going to be more efficient. We're going to extract the energy out of glucose really efficiently so we can get as much ATP produced as possible to power cell process. And so what we're going to do is oxidized glucose. What does that mean? In aerobic respiration, we are oxidizing glucose, taking away electrons. Very good. The final electron acceptor will be oxygen. The three major steps to glucose oxidation, we're trying to remove the carbon atoms efficiently, to remove energy efficiently. To make what? We're oxidizing glucose to make what? What are we trying to make? In the form of ATP. We're starting with glucose. We're going to pick off its electrons to make ATP. Okay? Right from, we're just going to phosphorylate ADP to make ATP. So we're starting with glucose to make ATP. But it's not a magic transformation. There's a very regulated processing of glucose before we can see any ATP synthesized. The three major steps to glucose oxidation under aerobic conditions. Okay? Under aerobic conditions, the first step that we're going to study is called glycolysis. In glycolysis, what does the word lysis mean? We use the word, that suffix lysis mean, to break down their split. In glycolysis, we're splitting sugar. Glyco, sweet, splitting sugar, okay? What that translates to in glycolysis is just splitting a sugar. And we're studying glucose metabolism, the breakdown. I'll just kind of give you the nitty-gritty of each of these first. Glycolysis is the splitting of sugar. And we're going to learn, if you read ahead, where does this process occur? In the cytoplasm. In the cytoplasm. Glycolysis, splitting of the sugar, occurs in the cytoplasm of a cell. Could be in a plant cell. Could be in a cell in your muscle, like in this image. Could be a cell in your lungs, wherever. Could be a cell in your dog's tail. Okay? Glycolysis can happen in the cell cytoplasm to split glucose. What it produces is a molecule called pyruvate. P-Y-R-U-V-A-T-E. Glycolysis splits glucose into two separate pyruvate molecules. Splits it into, how many 3. It splits 6 carbon sugar in half. So each pyruvic acid molecule in pyruvate must have 3 carbon atoms. So glycolysis occurs in the cell's what? Cytoplasm. Splits 6 carbon glycolysis into 2 3 carbon molecules called pyruvate. It's this 3 carbon molecule that is further oxidized or processed to release more. What are we trying to release? ATP. Well, we're cutting the glucose. We're using it up. So once we break it down, we're just going to keep breaking it down. Because we said we're going to do this slowly to be efficient. So let me start back over. Glycolysis is our kind of starting pathway, metabolic pathway that splits a molecule called glucose. Splits glucose to make pyruvate. Okay. This glucose is broken in half into 2 separate pyruvate molecules. That can be further broken down in the next step. But before we get to the next step, I want to finish talking about glycolysis. So yes, glycolysis splits glucose into 2 3-carbon pyruvic acid molecules. As well, it liberates some ATP. If you read ahead, how much ATP is produced? And how much net ATP is produced following glycolysis? 2 usable ATP molecules are produced. This is critical. Write the word net. Any 2 net ATP molecules are made following glycolysis. 2 net molecules of ATP are made. The word net implies what? You ever look at your check and you see your gross pay, your net pay after working? Yeah, so we're going to see a balance. So that word net is really important. And a familiar molecule called NADPH. A couple of molecules of NADPH are also released. So pyruvic acid, ATP, NADPH, and hydrogen ion. Oh man, not NADPH, it's NADH, sorry. Up on my photosynthesis. So the pyruvic acid, following glycolysis in the cytoplasm, pyruvate is made, ATP is made, NADH is liberated, and hydrogen ions are released. I'm sorry? What'd you say?
Liberated, what's that exactly?
Liberated, freed, released. Alright, so that's the crash course to glycolysis. Happens in the cytoplasm, takes a 3-carbon molecule called glucose, splits it in half to produce 2 3-carbon containing molecules called pyruvate. It also liberates or releases a couple of molecules of ATP, NADH, and hydrogen ions. In this pathway, we want to follow pyruvic acid or pyruvate metabolism because it has more carbon atoms and more what? More stored energy. Okay. And so, the next step in this aerobic respiration is to deal with pyruvic acid. And the name of this step is called the Kreb cycle. Okay? So the next major point to glucose metabolism is called the Kreb cycle. The Kreb cycle will metabolize. I don't have time to fully clarify, so I'm going to not state that what I'm going to state is not 100% accurate, but it's the shortest way to get there. Okay, and I'll clarify it all for you. The Kreb cycle will metabolize the byproducts of aerobic acid in the mitochondrial matrix. The Kreb cycle will metabolize, or we could say oxidize, a byproduct of pyruvic acid. In the mitochondrial matrix. This implies that pyruvic acid has to be transported where? To the mitochondria. So pyruvic acid actually has to get transported from the where? Where was it made? Cytoplasm across the outer and the inner membranes of the mitochondria into the matrix. Where then the Kreb cycle can take place. The Kreb cycle occurs in the matrix of the mitochondria. It also produces two slowly little molecules of ATP. I'm not going to give you the name yet. I'll give it to you on Thursday. You may know it. It produces two molecules of ATP as well. It probably also releases NADH and hydrogen ions. It'll release a whole heck of a bunch of NADH and their associated hydrogen ions. It also releases carbon dioxide, and I also heard another important molecule called, did I hear it whispered? Like this one. FADH. So in summary, the Kreb cycle occurs in the matrix of the mitochondria. It utilizes a byproduct, and it's called acetyl-CoA. Go into better detail on Thursday. It also liberates a little bit of ATP. Does it liberate any more than glycolysis? No. So at the face value, it doesn't look really advantageous. Unless we start to nitpick what else is produced. A lot more, 3 times more NADH, and another similar molecule called FADH2, which is carrying what? Transporting what? What do we said these things carry? Hydrogen ion and or electrons, right? Which have very similar to the role of NADPH. Electrons carry give energy. Okay? So Kreb cycle doesn't directly make a lot of ATP, but it does produce a lot of energy transferring molecules called NADH and FADH2. This cycle produces a lot of NADPH and another similar molecule called FADH2, where they can be used in the third phase called the what? The electron transport chain. So the electron transport chain uses NADH and FADH2 to do what? To transfer energy to make a whole heck of a lot of, what are we trying to make? A whole heck of a lot of ATP. So the electron transport chain utilizes those coenzymes NADH and FADH2 to make abundant amounts of ATP. And this is where oxygen is used. Oxygen is used at the end of the electron transport chain to accept electrons.